The United States Department of Energy, DOE, in a strategic move to secure technological and economic supremacy in the burgeoning global bioeconomy sector currently valued at over $4 trillion and projected to expand exponentially to more than $30 trillion over the next 3 decades, is deploying an unprecedented fusion of robotics, advanced automation, and artificial intelligence to fundamentally dismantle the traditional, painstakingly slow paradigms of biological research and usher in a new era of hyper-accelerated discovery. By commissioning and developing a suite of autonomous scientific platforms designed to systematically decode, manipulate, and harness the immense, yet largely untapped, metabolic potential of the planet's microbial world, specifically targeting anaerobic bacteria and fungi that thrive in oxygen-free environments and represent a vast reservoir of genetic and biochemical novelty for pioneering sustainable solutions to some of society's most pressing challenges in energy, manufacturing, environmental remediation, and national security. This visionary initiative is being realized through the creation of the Anaerobic Microbial Phenotyping Platform, AMP2, a first-of-its-kind, fully automated scientific infrastructure built by the synthetic biology company Ginkgo Bioworks and situated within the Environmental Molecular Sciences Laboratory at the Pacific Northwest National Laboratory, PNNL, in Washington State, which is slated to become operational in January 2026 and is explicitly engineered to function as the world's most capacious and advanced autonomous system for the large-scale cultivation, experimental manipulation, and phenotypic characterization of anaerobic microbes, thereby enabling a national cohort of researchers affiliated with the DOE to move beyond the historical constraints of manual, serial, and often speculative trial-and-error methodologies that have dominated industrial microbiology for centuries, and instead to engage in a massively parallel, robotically executed, and intelligently guided exploration of microbial physiology that will compress discovery timelines from years or decades into mere days or weeks. A transformative leap in throughput and insight that will serve as a critical functional prototype and technological proof of concept for an even more ambitious and expansive future facility known as the Microbial Molecular Phenotyping Capability, M2PC, for which groundbreaking is scheduled for the coming year with an anticipated operational date of 2030, and which together will constitute the planet's preeminent autonomous microbial research infrastructure, forming a seamless, integrated discovery engine that positions the United States at the absolute forefront of biotechnology, bioindustrial manufacturing, and the innovation of next-generation materials derived from biological rather than petrochemical feedstocks. The operational genius and transformative power of these platforms, funded through the DOE's Office of Science, lie in their deeply integrated cyber-physical architecture, where one layer of the autonomous science workflow involves a symphony of sophisticated robotic systems encompassing robotic arms, liquid handlers, automated incubators, and high-throughput analytics instruments, performing the repetitive but critical physical tasks of sample preparation, inoculation, media dispensing, container transfer, and environmental modulation on a relentless 24-hour, 7-day-a-week schedule that far surpasses human endurance and consistency, while a concurrent and deeply interwoven layer of artificial intelligence, comprising machine learning algorithms, computer vision systems, and autonomous AI agents dynamically analyzes the torrents of multimodal data including genomic sequences, proteomic profiles, metabolomic signatures, and growth kinetics, generated by these experiments in real time. Using this continuous stream of evidence to not merely report findings, but to actively formulate and test new hypotheses to recursively redesign the parameters of the very next round of experiments and to autonomously execute complex, branching experimental workflows where one investigation fluidly and intelligently initiates another without the need for constant human oversight or intervention, thereby creating a closed-loop, self-optimizing, scientist-in-the-loop system that can identify subtle patterns, correlations, and causal relationships across multi-dimensional parameter spaces at a scale, speed, and level of complexity, utterly intractable to even the largest teams of human researchers relying on conventional techniques. This paradigm-shifting approach, as elucidated by PNNL's Interim Director of Earth and Biological Sciences, Douglas Manns, addresses the core scientific challenge of disentangling the control levers of microbial behavior by providing a previously non-existent level of experimental technology that allows the DOE and the broader scientific community to systematically probe the genotype to phenotype map under a near-infinite combination of environmental stressors, nutrient conditions, and genetic perturbations, thereby generating the colossal high-fidelity datasets required to move from observational biology to predictive and prescriptive engineering of biological systems. A point emphatically underscored by Scott Baker, the microbial molecular phenotyping capability leader who frames the quest as a monumental effort to understand the foundational biology of microbial phenotypes, noting that science has scarcely scratched the surface of planetary microbial diversity, and that achieving a broad, predictive understanding of what these bugs do necessitates the confluence of a massive coordinated research community and one of the world's most sophisticated AI-integrated laboratories, a convergence he describes as a perfect storm of technology developments poised to make disruptive leaps in biotechnology for the bioeconomy. Consequently, these autonomous platforms represent far more than just advanced laboratory equipment. They are the core physical infrastructure of a broader national strategy, explicitly aligning with and powering the U.S. government's recently announced Genesis mission, a dedicated cross-agency endeavor to unleash an age of AI-accelerated innovation by constructing an end-to-end AI platform that will federate and harness the federal government. US's immense, decades-spanning collection of scientific datasets, the largest such repository in the world to train next-generation scientific foundation models and deploy purpose-built AI research agents capable of autonomously generating novel hypotheses, orchestrating entire discovery workflows, and ultimately propelling an exponential acceleration in the rate of scientific breakthrough, thereby ensuring that the United States can rapidly translate the latent capabilities of anaerobic microbes into tangible technologies for producing biofuels, bioplastics, pharmaceuticals, carbon-negative chemicals, and novel methods for biomining critical minerals, all while establishing an unassailable ecosystem of technological leadership that will define the trajectory of the 21st century bioeconomy and secure long-term economic competitiveness and national security.